Okay, so it is, you're gonna stand up. Okay. So it is my pleasure to introduce Tim Legare. I'm sure you all know him. He is the fish lab intern here at Archbold, kind of adopted by the Herp lab. And so Tim, a little about Tim. So he grew up in Florida, in Titusville. He's got his family in the back row back there. He did his undergraduate at University of Central Florida, and then he spent some time working with fish and some other organisms. Did some work with the Illinois Natural History Survey, doing some fish work, and then we actually took him from Hawaii, where he was working on some little fire ant work. And then since Tim has been here at Archbold, he has been doing a lot of work to help increase our understanding of fishes around Archbold. So he did some surveys out at the ranch even, so he was going back and forth a lot. He did some work with pygmy sunfish, trying to figure out how to keep them in captivity so that he can potentially do some work on them later. He did some work on the African jewelfish, which is an invasive species you'll hear about a little bit. And then he's done some work he's going to tell you about today. And for his exciting future, he is going to be starting a PhD program in the fall with Dr. Becky Fuller at University of Illinois. So we are very excited for him, and yeah, look forward to seeing what he's going to do in the future. So without any further ado, I'm going to let you hand it over to Tim and let him tell you about what he's been working on.
Okay. Hopefully everyone can hear me on Zoom. Okay, awesome. All right. Thanks, Jess, for the introduction. Thanks for coming, whether in person or online. My name's Tim Legare. I've been the fish intern here since October. I'm going to be presenting some of the work I've been doing while here at Archbold. The title of my project was Are the effects of salinity on predator sensory cue perception in the Eastern Mosquitofish? What are the effects of salinity on predator sensory cue perception in the Eastern Mosquitofish, Gambusia holbrooki? So, there we go. Globally, the effects of climate change are becoming impossible to ignore. In Florida, this is especially true. We're beginning to see saltwater incursion events last longer and occur more frequently. Many fish species are unable to tolerate the shifts in salinity that occur with these events. Seawater is on average 35 parts per thousand. Most freshwater species struggle significantly to handle a 5 parts per thousand change. The result is usually large-scale mortality. These more frequent saltwater incursions will cause a drastic shift in community composition. For the species that can tolerate and remain, how will their behavior change in response to new environmental conditions? Will maladaptive behaviors arise, compounding already decreased fitness? The fish I use for this study are able to tolerate a wide physiological range. They're an ideal system to explore the possible outcomes of behavioral shift in altered ecosystems. If physiologically tolerant prey species show a loss in ability to express proper anti-predator behaviors, it implies much worse for our less tolerant, less monitored, less profitable aquatic species. For my project, I was interested in understanding exactly how saltwater stress may impact freshwater fish behavior in response to predator cues. Specifically, I examined the relationship between salinity stress and predator cue recognition in Eastern mosquitofish. Salinity can impact memory, stress levels, and olfactory sensitivity in fish, and thus may influence behavior and predator-prey dynamics. It's not difficult to imagine how stress can impact behavior. I'm sure many of you have seen me pacing up and down the station today. My behavioral response is increased activity when exposed to the stress of a research presentation. Hopefully, it leads to an increase in fitness. I'm particularly interested in the predator-prey interaction of mosquitofish and the African jewelfish, a common invasive species throughout southern and central Florida. I chose to use mosquitofish and jewelfish for a few reasons. They're both plentiful on archable property, both have an interesting body of literature to build on, they both have very high physiological tolerance, including salinity. Before I discuss specifically what I did in my project, I'll talk about each species individually. This will hopefully provide some context about why I find this system interesting and what influenced this study. This is our predator species, Hemichromis letterneu, or the African jewelfish. As you can see, it's a really pretty fish. It's likely they were released in Florida by Aquarius. They were first collected in Hialeah Canal in the 60s. Since then, they spread west and north at a remarkable pace. Unfortunately, we have them all over archipel property. If you put a minnow trap out on station, this is predominantly what you're going to find and at high density. They're originally from West Africa, with populations also occurring in coastal basins of Cameroon, the Democratic Republic of the Congo, and the Nile Basin. They're well adapted to savanna wetlands and are recorded in brackish habitats throughout their native range. They're egg layers, monogamous, and exhibit parental care. In Florida, they occupy a similar niche to our native sunfish species. They've been shown to disrupt their egg laying and nest building. Everglades populations have been experimentally shown to suffer no effects on growth or reproduction, up to salinities of 50 parts per thousandth, and they can tolerate all the way up to 60 parts per thousand, albeit with quite a bit of mortality. Just a reminder, seawater is on average 35 parts per thousand. They're capable of tolerating very low dissolved oxygen and a pH of below 3. Currently in Florida, the only thing slowing their spread seems to be temperature limitations. This is our prey species, Gambusia holbrooki, or the Eastern Mosquitofish. This is a melanistic male, but I thought it made for a better picture. Mosquitofish have different life histories than jewelfish, but share the abnormal ability to tolerate extreme physiological change. For starters, they're live bearers without parental care. Like jewelfish, globally they're considered invasive. They're fin eaters and have been responsible for collapsing populations of small-bodied native fish worldwide. Fortunately, they're native to Florida. In terms of physiological tolerance, they can withstand salinities up to 36 parts per thousand, albeit with some mortality, pHs below 3, and low dissolved oxygen. Importantly, they've been shown experimentally to have their behaviors moderated by changes in salinity. A study in Italy has previously showed that mosquitofish aggressiveness towards an endangered native cyprinodont was being hampered by salinity, and that food competition was decreased. The cyprinodont is becoming isolated to brackish areas of its range due to this abiotic mediation of mosquitofish. So the first question I asked about this system was, does an increase in salinity cause a change in mosquitofish anti-predator behavior? The inherent stress of saline conditions may result in abnormal prey behavior. Salinity has been shown to affect cue perception. Will anti-predator behaviors be displayed at all? There's the possibility of novel behaviors being observed. The majority of known anti-predator behaviors in mosquitofish come from studies conducted in freshwater. Are mosquitofish already capable of undescribed behaviors that arise only in saline environments? Finally, the inherent stress of salinity may make anti-predator behaviors overly costly for the fish in terms of energy expenditure. Will fish ignore predators, instead choosing to save energy in the event of an attack? My second question was, do Eastern mosquitofish show a stronger response to either olfactory or visual cues? Previous work has suggested that in the predator-prey relationship of jewelfish and mosquitofish, that mosquitofish are relying predominantly on olfactory cues for predator recognition. Jewelfish are an ambush predator. Olfactory cues alone may signal more danger than being able to see the jewelfish itself. Will mosquitofish respond more strongly to olfactory cues as they signify a danger unseen? My third question was, does salt stress change mosquitofish anti-predator response to visual or olfactory cues? Salt water may interfere with the ability to perceive olfactory cues. Will there be a change in reliance between the olfactory and visual cues? In murky water conditions, being unable to rely on olfactory cues may result in significantly increased predation risk. Forced reliance on a single cue may make fish more vulnerable to predation. So to answer these three questions, I first needed fish to experiment on and to ensure their salinity tolerance aligned with the literature. We performed preliminary salinity trials with small-scale collections from around Archibald. Turns out that jewelfish at Archibald are showing a significantly decreased salinity tolerance to those down south. However, the Archibald mosquitofish show similar salinity tolerances to those predicted in the literature. At 20 parts per thousand, we can assume that mosquitofish are physically stressed, however, still capable of eating when fed and behaving normally with conspecifics. We see small-scale mortality if mosquitofish are left too long at 20 parts per thousand. After ensuring that we have fish here that can meet their criteria needed for my experiment, I began collecting in mass and randomized the collection sites in outdoor mesocosms. All mosquitofish collected were from bodies of water that had jewelfish. It can be assumed that the mosquitofish are familiar with jewelfish as a predator. Even if jewelfish introductions are new, prior research has demonstrated that naive mosquitofish recognize jewelfish as a predator, even by olfactory cue, and behave accordingly. So I collected over 300 mosquitofish. It's actually a lot more than that, but I collected over 300 usable mosquitofish. To answer my questions about how salt stress influences anti-predator behavior. how different predator cue types interact with salinity. I divided fish into either visual or olfactory trials. I then further divided fish into saltwater or freshwater treatments. This resulted in 13 groups of 6 fish for each treatment combination. I only used female or juvenile mosquito fish of similar size to prevent the distraction of mating and the behaviors that coincide. Within each cue block, olfactory and visual, the trial timeline looked like this. Fish were further divided into saltwater and freshwater. Fish in the saltwater block were first acclimated in mesocosms outside, where they were brought up to 20 parts per thousandth over the course of a week. Prior to trials, they were fasted for 24 hours, and then brought into the lab at a minimum of 12 hours before the experiment to acclimate to the laboratory environment. Freshwater fish, both mosquito fish and jewelfish, were maintained outdoors as well and treated the same. Groups were alternated to which cue they were exposed to first, and between trials, they were maintained in the lab. They were fed after a trial, given a day to metabolize, and reran 48 hours after their first trial. For olfactory cues, we made a cue solution based on a prior study. This was created by overstocking a tank with either jewelfish or mosquito fish, allowing them to soak for 3 days. We removed the water, which was then frozen. Before a trial, we defrosted the cue we created. Visual cues were an individual jewelfish or mosquito fish. Unique fish of similar size were used for each trial. So these are the experimental tanks. You can see that we have two tanks facing each other with a camera watching the side and a camera covering the top-down view. We have yarn tied to a piece of fishing line in the middle of the tank that functioned as structure and simulated vegetation. It's important to note that it occupies the whole water column. This is to prevent a depth bias later. In our second tank, we can see a barrier is being used to create a subdivision. For visual trials, this ensures that cue fish remain in proximity to our experimental fish. For olfactory trials, cue was delivered from the same point. The mark's not in the photo, but this is approximately where it was. At the start of a visual trial, this piece of paper was removed to reveal the cue fish. At the start of olfactory trials, the clapping motion was done inside of the cameras to create a similar level of disturbance and for later syncing of the video. The trials were conducted by first placing a group of 6 mosquito fish in the experimental tank and allowing a 10-minute acclimation period. For visual trials, the cue fish were also given the same 10 minutes. After this acclimation period, the cameras were started, and either the paper was pulled away to reveal the cue fish for visual trials, or 10 milliliters of cue solution was pipetted in for an olfactory trial. The room was left, fish were given 30 minutes. After video was collected, they were either fed or euthanized for measurements, depending on what stage they were at. Still images were pulled at 1, 5, 10, 20, and 30 minutes from each camera. They were moved into Lightroom to increase contrast, and then measurements for variables of interest were taken. So we're going to look at some of those images now. So this is an image from the side-facing camera. There are 4 visible mosquito fish, believe it or not, and a jewelfish, because this was a visual trial. From this, we took depth for all the visible fish. So depth is one of our variables of interest, but it'll also be important later for calculating a Euclidean distance measure. By taking the mean measurement from each group at each time point, the effect of lost fish in images is decreased. This is our top-down view. We can still see our 4 mosquito fish and jewelfish. These red lines are the distance measurement, and these yellow lines are the distance from structure measurement. Using the 2 red line measurements, what's depth from the last photo and the distance measurement from this photo, we can calculate a Euclidean distance measurement. So with depth from the side image, distance from the top, you can find the Euclidean distance, which I'm just going to refer to as distance going forward. So now we have a way to measure the 3 variables of interest: depth, distance, and proximity to structure. Now that we have our data, we need to analyze and interpret. We'll start with some predictions. We predicted that there's going to be a strong behavioral response to olfactory cues. Prior research suggests mosquito fish are predominantly relying on olfactory cues to identify jewelfish as a predator. We expect that distance from cue origin, when compared to the control, is going to increase in freshwater. In saltwater, distance is expected to show no change. Mosquito fish have been described moving to the upper water column after detecting jewelfish. Predict that mosquito fish will occupy the upper water column in freshwater and exhibit no change in saltwater. In freshwater, I expect them to maintain their distance from structure. In saltwater, I suspect structure use might increase. The differences that I'm predicting between fresh and salt water is because I suspect that mosquito fish will be unable to detect predator cues or will show decreased perception. To determine if salt stress influences anti-predator behavior in response to predator olfactory or visual cues, I used a linear mixed effects model for both olfactory and visual trials. The mixed effects included Q, which was the jewelfish or mosquito fish control, water type, which was 20 parts per thousand salt water or freshwater, time and the time of measurement, which is 1, 5, 10, 20, and 30 minutes. There's also the repeated effective group. Each group was tested for both cues. First, we'll look at the distance from olfactory cue origin. We have our distance from Q on the y-axis and our freshwater and 20 parts per thousand treatments on the x-axis. Control Q is gray and predator is red. Water and Q did not seem to have an effect. This does not align with our predictions, but it does make sense. As the Q disperses, it is going to become detectable through a majority of the tank. Interestingly, the lack of response in relation to the Q origin suggests that while the fish may be able to detect the Q, pinpointing the origin may be impossible. Of course, this interpretation assumes that there's a gradient of Q intensity from the origin outwards. It's possible that when we reanalyze this data as a time series, we may see an aversion to Q origin at earlier periods in the trial. Increased activity may also be a better predictor than distance from origin. Here we are looking at the average depth during the olfactory trials. The y-axis is flipped here and now corresponds to depth, so higher up on the y-axis is closer to the water surface. Here I found significance between the water treatments. Fish were on average shallower during our salinity trials. This was true for both predator and control cues. It's likely this response is due to stress from the salinity. However, movement closer to the surface is a known successful anti-predator response to jewelfish. So even though the Y of the response is wrong, the action taken is correct and minimizes risk of jewelfish predation. Finally, we have a distance to structure throughout our olfactory trials. I didn't see a strong response for any of the mixed effects. As fish move through the tank, their position relative to structure is going to change. My predictions for freshwater are correct by omission. We don't see close proximity to structure, and being significantly far away may be limited by tank size. I was wrong, however, about the saltwater treatments. I don't see any increase in structure proximity. So let's see how well the results match the predictions. In freshwater, I predicted an increased distance from Q origin when compared to control. This wasn't observed. In saltwater, there was no change in comparison to the control, is what I thought. The freshwater depth measurements met my predictions. Fish remained towards the upper middle portion of the water column. There was no change predicted for saltwater. However, we saw fish come higher across both the control and predator treatments. This is likely, like I said before, a salinity stress response, but this also happens to be the ecologically correct anti-predator behavior. I was wrong for my predictions for structure use across both water treatments. We don't see significant distances being maintained in either direction. Importantly, the lack of significant closeness suggests the mosquito fish are choosing to forego structure use in stressful environments or when presented jewelfish signals. Now we'll begin looking at the results of the visual trials. Visual systems provide a different set of information than olfactory. Fish are able to identify exactly where threat is coming from, water clarity permitting. So I expect to see a behavioral difference from the olfactory trials. I expect that distance to the predator cue is going to be increased from our control in both fresh and salt water. I predict that in both water conditions, mosquito fish will respond by remaining close to the surface. If mosquito fish can see the predator and identify that it is not in the structure, they may attempt to use it to remain out of sight. Across all of the variables, I expect to see the same response behaviors between salt and fresh water. Unless salinity causes changes to the visual system itself, fish should be able to respond equally across treatments. So we're going to first look at the distance from visual cue. Once again, we have the distance from Q on the y-axis and the freshwater and 20 parts per thousand treatments on the x. Control Q is gray and predator is red. These results align well with the predictions. Q is a significant predictor, finally, while water is not. Mosquito fish are remaining further from predators in comparison to the control. Similar response across water treatments implies that the inherent stress of saline conditions does not elicit a change in response. The intensity appears the same. Moving on to the depth visual trials, note that once again the y-axis is flipped. We see no significance in water or Q. We are seeing a trend towards significance for water. Like many of our results, there is more to be gleaned from what is not being shown. While depth appears important when stressed or detecting the olfactory cues of an unseen predator, it appears to become less important when the predator is in sight. Instead, the total distance maintained seems to become much more important. This doesn't fit our predictions, as we assign equal importance to decreasing depth and increasing distance. Finally, we have the distance to structure. As a reminder, I predicted close proximity across both water treatments. We see no significance across Q or water. We got about the same response as our olfactory trials. Structure doesn't appear favored, and true aversion may be hard to detect. We can determine that even if a predator is visible, structure use does not seem to increase. So a quick summarizing of the results of the visual trials. We see the expected increase in distance compared to control and across water treatments. We do not see the results I predicted for depth or proximity to structure. As I said earlier, for depth, the importance of position in the water column appears to matter less when the predator is seen. Decreasing the distance to the surface is safer when a predator is unseen, and lateral movement could result in swimming directly into it. These results also suggest that even though the predator is visible, the distance is not being sacrificed to instead seek cover and shelter. It's possible that even if the predator is visible, structure offers the risk of hidden predators. Even without detectable unseen cues, there still seems to be an aversion. So how do these results fit into my questions? Just a reminder, the first question I asked was, does an increase in salinity cause change in mosquitofish anti-predator behavior? My results show that saline environments induced a change in depth preference. In olfactory trials, mosquitofish chose to remain shallow. This was not in response to cues, but it did align with expected anti-predator behavior. In saline conditions, it's probable that mosquitofish will respond by moving out of the expected range for jewelfish predation. In visual trials, we saw that they remain deeper. However, this is more likely due to the positioning of the jewelfish itself. Increased distance appears to be favored. We can conclude that salt stress may change behavior. However, there is no evidence that these changes are actually maladaptive. My second question was, do mosquitofish show stronger anti-predator response to either olfactory or visual cues? The results show that visual cues were the only type to elicit an anti-predator response in mosquitofish. This disagrees with previous research. Importantly, salinity did not affect the response to visual cues. My results suggest that mosquitofish are relying more on visual cues than olfactory to determine predator risk. Single cue reliance is ecologically dangerous, as change in water conditions, like clarity, put prey at an increased predation risk. My final question was, does salt stress change mosquitofish anti-predator response to olfactory or visual cues? I found no interactions between cue and water type. Mosquitofish only showed response to water type or cue individually. This suggests that mosquitofish may rely on general anti-predator behaviors when risk is known versus unknown. Do not adjust the behavior due to salt stress. So some concluding thoughts. We do see behavioral abnormalities arise in saltwater. However, they do not appear maladaptive. We will need to do further research to explore how sensitivity is affected through time. Even without maladaptive behaviors being displayed, the abiotic change facing Florida is going to greatly impact species composition. The mosquitofish do not seem like they'll have their behaviors impacted by this change in an ecologically meaningful way. However, this assumes they're able to maintain tolerance to chronic salinity. We can take cautious hope that other physiologically tolerant fish will be able to maintain themselves in changing ecosystems, hopefully mitigating even further potential loss of species richness. There's still more work needed to be done. The prior work I've built this study off of was conducted with direct observer scan sampling. I've added recorded scan sampling with quantifiable data. I believe that these scan sampling experiments lack useful resolution and limit the behaviors that can be measured. I intend to reanalyze my videos using deep lab cut produced by the Mathis lab to track fish across the whole 30-minute trials. This will allow us to examine some behaviors we couldn't previously. We'll be able to quantify changes in activity, total distances moved, proximity to conspecifics to determine shoaling, and quantify predator inspection behavior. I've already been working on adapting my data for deep lab cut. The GIF you see on the right was created with about 120,000 training iterations. I'm excited to re-examine this data set with a higher resolution method, as I think it will allow for better analysis of these trials and better answers to my questions. So with that, just some acknowledgements. Thank you everyone for coming to the talk, either in person or on Zoom. Thank you to my funders and Archbold for this opportunity. Special thank you to the Fitzpatrick Lab for their helpful comments and support. Thank you to Jess and Caitlin for being excellent lab mates. Thank you to Betsy and Rachel for having us in the herpetology lab and for your comments. I want to thank the current and past intern cohort. I've been fortunate to live and work with a really great bunch of people. This experience wouldn't be half what it is without you guys. And as this is kind of my first and last presentation since undergrad, and I'll be going back to school this fall, I just want to give a special thanks to the folks I got to work with at UCF, the INHS, and MISC. Without the experiences I gained there and the mentors I had, I'd be half the scientist I am today. And finally, a special thanks to my family for their support and instilling an interest in biology from a young age. I really appreciate the support that's gotten me this far, and I'm always looking for opportunity to collaborate in the future. So, any questions?
Okay, I just want to say I see a lot of these names online, just so they were watching. Right, I have the microphone if anyone has a. Right, I thought you Tara first, sorry.
Can you describe what the olfactory cue is like for fish? Like, what kind, does it have particular smell?
Yeah, basically. So, I guess that's a good way to describe it. In fish, there's olfactory cues that can do quite a few different things. So mosquito fish, there's a lot of work done with alarm cues. So if a mosquito fish is injured, you know, fluid, blood enters the water, and other mosquito fish are able to detect that, and they will respond by, dispersing or, trying to get away from whatever the perceived threat is. So there's a lot of that kind of response to cues, even if there isn't damage like that. So in predators, there's a specific chemical in their guts usually, and the mosquito fish can smell that. And to them, it says, this is a predator, stay away. They can also detect, conspecifics in the guts of their predators. And so, yeah, it's basically, I don't know, like a way to relate it to like mammals, but yeah, you can smell if something's dangerous, basically.
Okay, there's a few questions on Zoom. One is, can you explain more of how you conducted olfactory cue for the fish?
Sure. So, like I just kind of briefly mentioned, and then we can go back a little. Sorry. As I just briefly mentioned, mosquito fish are able to detect damage to conspecifics through olfactory cues. So we're not really interested in that for these trials because we know there's going to be a behavioral response that's going to signify I'm in danger. Was this on Zoom, I assume?
Yeah.
So to create so we want to use mosquito fish as a control for our treatments. So we want the water that we're pipetting into the tank to smell like just happy, healthy mosquito fish. However, for jewelfish, like I kind of briefly mentioned, they have innate cues that signal to mosquito fish that they are a predator. And so we want to get those innate cues from the jewelfish. So we just put regular jewelfish in some water, like let them sit in there for like 3 days, and then take that water out, and it should have remnants of those smells. And then yeah, pipette it into the tank and then look for a response. I hope I answered that well enough. Sorry.
Question from Sarah Sinise. Is it possible natural populations of mosquito fish taken from seasonal ponds have inherent anti-predator responses that are stronger than bred mosquito fish lines? How would that fit into your results? And although you're a haline, how would the initial salinity content of their home pond influence results? That was a lot of questions there.
So the first question was difference between wild mosquito fish and raised mosquito fish, I believe.
Right.
Right.
Yes.
So for this, these are all wild. They're not raised. I'm sure there's differences, but in terms of, increased saltwater incursion in Florida. I think basing the research off the wild is probably best bet. And then there was the
how would initial
ponds.
salinity content of their home pond influence results?
Yeah, so we'd expect it to, most likely. So the mosquito fish out here, they're in 0 parts per thousand saltwater. They're able to tolerate that 20 parts per thousand, but not for long. They do eventually suffer some mortality. But that's kind of exactly what we're interested in. These fish, we want to know what these fish are going to do when they're taken from an environment where they're naturally in saltwater and then put into a more saline, more saline abiotic condition. And so, yeah, I guess that would be my answer. Yeah, local adaptation does exist. I'm sure the mosquito fish, probably in the Everglades, have a higher natural salinity tolerance just from constant exposure. I'm not even sure if that's really true, to be honest, just because, yeah, prolonged stress is really never good for any organism. So,
if mosquito fish as a species move higher in the water column with increased salinity, this is from Linda, by the
okay.
way, will that mean they will be less available to jewelfish, or is the difference in depth not sufficient?
Yeah, so they should be less available to jewelfish the higher up they are. Like I said, jewelfish are a ambush predator. They seem to have a really strong aversion to going up in the water column. If you keep them in a tank, they mostly just lay on the bottom and look very drab, and they try to hide in any kind of little piece of, vegetation, or if you have I put pieces of PVC, they like to hide in that. I think they're very averse to going towards the surface because of bird predation. And so the mosquito fish, a lot of bird species are not really going to bother with a mosquito fish. While with a jewelfish, something like an osprey is probably going to be pretty happy to grab you. So
the Maui invasive species group says, look at the big brain on Tim.
yeah, I assume. Thank you, or Monty. Thank you.
Locke says, in the wild, both the predator and prey are going to be exposed to the same salinity challenge.
Right.
So while it is reasonable to only focus on the prey species for the experiment, do you have information about how salinity affects the jewel cichlid?
Yeah, so I didn't plant that. I'm sure a lot of people probably think I planted that. I didn't plant that. Yeah, so I did some preliminary salinity trials because the first part of this experiment, and there's 20 slides somewhere talking about it that got cut. First part of this experiment is, do you see a change in predation anyway? So does this really matter? And so I tried testing salinity tolerance in the jewelfish here on station, and it's actually incredibly low. They're supposed to be able to withstand 20 parts per thousandth direct addition to water, and they just don't do that here. In the Everglades, they survive at 100%, 100% rate over the course of a week, and at Archibald, it's 0%. And so I've gotten some permits. I'm going to go throughout the state and collect more jewelfish from a mix of conditions and try to see why the Archibald jewelfish have lost that salinity tolerance. And is it like a phenotypic plasticity kind of thing, or a rapid adaptation?
How do you think this finding would apply to species less adaptable to salinity change, since these species are highly tolerant?
Yeah, so my suspicion is that they're going to show, a lot more stress response and a lot more stress behaviors. And so if you're already barely hanging on, and now you can't recognize predators, you're not really hunting for food correctly and all that kind of thing, your fitness is going to decrease even further. And it doesn't really matter that you might just be at the cusp of, tolerance if you can't, complete normal function. So we'll probably see die off.
Okay, any questions from
Unfortunately.
in here? Okay. Also, Becky said, nice job. Aaron David in the plant lab said, great job.
Thank you.
Sarah said, great job.
I will also say, Tim, great job.
Thank you.
Like setting up an experiment that setup is really, really cool. Breaking it down into all those protein groups
you.
of everything. That's fantastic. So I have just a quick couple of questions. One is, did you do an interaction between where you added both visual and olfactory cues?
No, so there's been a look at, the combination of cues and mosquito fish before. I think with pumpkin seed sunfish or something like that, or warmouth, I don't remember. And yeah, it looks like if you have two cues, they tend to rely almost a little bit more on one, but as conditions change, they're gonna change their reliance. So there does seem to be a bit of a Q hierarchy. The first experiment where I wanted to just look at predation risk was kind of my idea of combining Qs, just because you know two birds with one stone. You gain the data from are they getting predated at a higher rate, and if they're getting predated at a higher rate, you can assume that they're not using both Qs combined, in a way that's really beneficial or ecologically beneficial.
Yeah. Yep. Cool. And one last question: is there something physiologically good about moving up in the water column when the salinity is high for the mosquito fish?
That's a good question. I actually don't know. From what I understand about saltwater and mixing, I don't think there's any sort of benefit. Mosquito fish do have this kind of weird response to stress or like predators where they tend to come up really high. And they'll even like try to jump out of whatever tank they're in to like something else if they think it's going to be better. So like if you have, two tanks next to each other, they'll jump into the other tank, like, if they're getting chased by a male or something, or like getting harassed. So I don't know if there's sort of a similar response, but I mean, if you're in a lake or like a river and it gets really saline and you jump out, that's really not going to help you too much.
Cool. Thank you so much.
Yeah, no problem. Yep.
Oh, sorry.
Nice job, Tim.
Thank
I
you.
had a question about the breadth of your sampling around the station.
Yep.
Because we had done some surveys a few years ago of mostly seasonal wetlands on the station, and I was concerned about the extent to which the jewelfish are, colonizing these systems at Archbold. I seem to remember we only found them in a small proportion of the seasonal ponds. Do you feel like maybe they are?
Yeah, I've caught them in
more
quite a
common?
few. So, I use this little GIF just to be funny or whatever. And this is the sinkhole pond. They're not in the sinkhole pond, luckily, because it seems to be when they're not in a pond, the top minnow, like killifish populations are a lot higher, and so are the pygmy sunfish populations. But yes, some of the seasonal ponds definitely have them. The ones behind the rosemary bald, like you have to walk and go all the way through. That far one has jewelfish that I've caught all over the reserve and basically any of the water out there.
That makes sense to me because like so many ditches that would connect a lot of those wetlands and allow them to spread. But I'm kind of hoping they're still not in. There's still some that are a little less. They're a little less able to reach.
Okay.
Thank you.
Yep.
Cool.
If you want.
I had a question about like, I know the original solution you put it in that has like the olfactory cues from the predator, the jewelfish. I was wondering if you had like more jewelfish in there, would it increase like the amount of like the concentration of whatever is detecting, and would that affect the behavior? Would it like you show an increased response to that behavior if was the case, or is there like a threshold?
Yeah, so there's a prior study that had been done on jewelfish and mosquitofish interaction, and so I followed how they made Q. I actually did it at kind of a stronger concentration just because I wanted to ensure there was a response. In terms of I don't know any way to measure like how much is really in a solution rather than just putting the same amount of fish in and then putting the same, taking the same amount of water out and kind of hoping that you get a pretty good average. I will say I believe I used 5, either 5 jewelfish or 5 mosquitofish. I tried to use, representatives that were going to be similar to what I was going to do in like a visual trial. So, if I'm going to use a fish for a visual trial, I'll try to use a similar size fish for the olfactory trials. But it is kind of hard to compare or to understand how much Q is really in the solution between jewelfish and mosquitofish, just because, mosquitofish are very small, jewelfish tend a lot larger. And so by mass, jewelfish Q probably has more jewelfish or whatever you want to call it in it. Yeah. Yeah, I don't know a way to measure it. Thank you.